welcome you again, and let us hope that the shortcomings that were seen in the beginning will not hamper the presenter and the presentation in any way. We are extremely delighted, but as you know, technologies can sometimes create small problems. So there is a possibility, though we try to have only the people who are to be working on this to be logged in, but there is sometimes a problem of others logging in. We have not yet made provision to keep all others logged out. So I hope our Honorable Vice Chancellor has already spoken. Sir, have you given your opening remarks?
Not yet. No, not that. It does not hurt.
Okay, then we restart. Yeah. So it's an action replay, and it makes everybody excited to know what they are going to hear in the next 45 minutes from Dr. Vinod. And therefore, to keep their excitement patent, I request you to give your opening remarks, and then, of course, we will be introduced to all those who have logged in, and then after a brief introduction by Dr. Karunasagar. So may I request you, sir, to give your opening remarks?
Thank you, Indrani. Good afternoon and good morning to Vinod. I congratulate him and thank him for accepting our invitation to participate in this very interesting webinar program. As all of you know that we are in the COVID-19 crisis and the whole world is worried and concerned about this novel coronavirus. And as a health university, we are doing all our efforts to educate not only our teaching, non-teaching staff, everybody about this COVID virus. And the program conducted by NUPSAR is really appreciable, and I congratulate Indrani Karunasagar and Nitte Karunasagar and Anirban for organizing this very interesting webinar on COVID-19, and probably they're discussing about the effect of genetics on corona. As you all know, there is a heterogeneity in this effect of corona in different countries and different ethnic group of people. And even in India, we had upsurge of this virus, and even mortality is very high in Gujarat and Bombay and certain places compared to Kerala and other places. There must be something. I understand that there's got something to say about genetic presentation and its effect on COVID infection. I request Vinod to enlighten us on his study and what is observation, and so that definitely the people who are participating in the webinar will be enlightened by his view on the effect of or the role of genetics on this viral infection. I thank once again for giving this opportunity to give the opening remarks. I request Vinod to continue with this talk on the effect of or the role of genetics in COVID virus.
Thank you, sir, for your kind words and encouragement to these kind of sessions that help many scientists to enlighten themselves. Can I request Dr. Idya Karunasagar to introduce Dr. Vinod to all of the audience who, of course, would be very happy to hear about this young gentleman who is making waves.
Thank you very much. And indeed, I am very, very happy to introduce Vinod to all of you. He is one of the very bright young scientists who have come out of our College of Fisheries. So he did his master's degree in fisheries, and then he moved on to the Hanover Medical School, where he did his PhD. And from fisheries, he changed over to genetic susceptibility to infectious disease. And after his PhD, he continued there for some time in the Hanover Medical School in the clinical research group, and he was awarded the Riken postdoctoral fellowship from the government of Japan. And this is a very special fellowship which is awarded to foreign researchers with creative ideas. So he took up this fellowship at the Institute of Medical Science, Tokyo Medical School. And after his postdoctoral fellowship for 3 years in Japan, he came back to Europe. So he joined University of Groningen, University Medical Center, and he was a senior postdoctoral researcher there for 3 years, and then he was awarded a faculty position. So he became assistant professor in the Department of Genetics. And in 2017, he became the principal investigator and Hypatia tenure track fellow at the Radboud Institute of Health Sciences. So he got a research award of 1 million euros to carry out research on systems genetics approach to infectious disease. So his research in this area has been recognized internationally, and he was awarded the 2018 International Sepsis Forum Award, and he also delivered the award lecture at the European Congress of Clinical Microbiology and Infectious Diseases. And he has supervised already 7 international doctoral students, 2 postdoctoral researchers, and 6 master's students. And his publications, I think it will be very interesting for our young people to listen. He has already 60 scientific publications, 30 of which are in journals with more than 10 impact factor. So he has 2 papers in Science, 3 in Cell, 4 in Nature Medicine, 1 in Nature Genetics, and 2 in Lancet Infectious Diseases, and in Gastroenterology, 2 Nature Communications 2. So this can be envy of any scientist to have such a publication in such journals at such a very young age. And he has over 3,500 citations, and average citation per publication is more than 55. So it's really, we are very fortunate to have Vinod with us this morning to deliver this webinar on functional genomic approach to dissect the COVID-19. Over to Vinod.
Well, thank you very much, Dr. Karunasagar sir and Madam, and Dr. Satish and Dr. Aniban, for your kind introduction. I'm overwhelmed by the love and affection and enthusiasm, for my presentation. And good afternoon, everyone. So, without any further delay, I will just move us on and get us on with the presentation. I hope it works without any technical glitch. Could you please confirm whether you can see the screen?
Yeah.
Yeah, now we have the screen.
The screen is on. Okay.
All right. So I will.
Full screen.
So just for the.
Vinod, could you please put on slide? Yeah.
Yes. So now it should be fine. So once again, thank you very much for the kind invitation and introduction. So it is my great pleasure to present to you this webinar, which focuses mainly on the application of functional genomic approach to understand factors that contribute to COVID-19 disease susceptibility as well as severity. Now, COVID-19 doesn't really need an introduction or explanation now, as both the television and social media is literally flooded with the latest research and information about this disease and the virus. So instead, I only want to stress the fact that in less than 6 months, it killed nearly 500,000 people worldwide and is changing the world at many levels. Now, this pandemic is a global crisis, creating severe disruptions across the economy as well as health system. So therefore, getting insights into how to better understand and treat COVID-19 are desperately needed. Now, if we look at the worldwide epidemiological data, you may have already read this in news that more than 5 million people are infected by this virus. However, fortunately, more than 2 million people have recovered without any serious complications. And unfortunately, about 350,000 people have died. So this clearly shows that there is significant heterogeneity, both for susceptibility, so who gets the infection, who doesn't, even though they're all exposed to the virus, and also the severity of the disease. Once you become infected by the virus, the severity may vary depending on many different factors. Now, the important question is, which factors actually contribute to these differences? Of course, we already know some of the factors, some of the host factors, such as old age, gender. Men are more likely to die because of this virus than women, and other underlying health conditions such as diabetes or immunodeficiency and so on. Even though if we are able to match for all these factors, there is still significant heterogeneity among population in susceptibility and severity. So, as you may all already know, for any infectious disease, the three main factors that contribute to variability can be broadly grouped under host factors, pathogen factors, and environmental factors. So, as you can imagine, the dynamic interactions between these three major factors is what finally determine the disease phenotype, including COVID-19 disease susceptibility and severity. Now, understanding the implications of all these three factors is extremely important. However, for this webinar, I will focus on host genetics, mainly because, from evolutionary point of view, we know that pathogens have played a tremendous role in shaping our genome. So therefore, identifying genetic factors that contribute to COVID-19 disease susceptibility as well as severity could help in clarifying underlying biological mechanisms, which are important in developing novel drugs, as well as to make use of existing drugs, so-called drug repurposing. We can also use these genetic markers to identify individuals who are at unusually high risk, so that we can then provide a specific care and follow them up. And knowing genetic risk markers and risk genes will also help us to come up with personalized treatments based on their underlying genetic defects. Now, although we expect a significant role of host genetic variation, thus far, no studies have identified any particular genetic locus to be associated with COVID-19. Now, instead, there are suggestions and predictions on potential candidate genes, based on their known previous function in other infectious diseases or other viral diseases. And some of such genes you can think of are many genes from our HLA locus, where it codes for several alleles, and these HLA alleles are basically responsible for recognizing the viral proteins. So in one study, they identified HLA-B allele may influence binding efficiency of viral peptides, so thereby influencing the disease severity. And there are also other genes called mannose binding lectins, or IFIT3, which codes for an interferon transmembrane gene. So among these genes, one important gene that I would like to mention a bit more in detail is ACE2, you may all already read about this, that this is a receptor for coronavirus 2. And this protein is expressed mainly on epithelial cells, lung epithelial cells. And that's one of the reasons why we have such a severe phenotype with the infection of this virus in the lung. And interestingly, this gene is encoded by X chromosome. So it's mapped to X chromosome. And we know that there is threefold higher mortality in males compared to females. So it is very interesting to speculate that somehow having two copies of these genes, so female have two X chromosomes. So in that sense, these chromosomes may contribute to differential regulation of ACE2 protein, which may explain the differences in mortality between males and females. Well, this is a speculation. This needs to be investigated further. So traditionally, genome-wide association studies is a technique that's being used to identify genetic factors for many complex human diseases. So this is an unbiased approach where you collect thousands of DNA from thousands of patients and thousands of controls, and this DNA will be hybridized on a DNA SNP array, single nucleotide polymorphisms array, and then you compare the frequency of these polymorphisms between cases and control to basically look at association between these polymorphisms and the disease phenotype. Now, for COVID-19 as well, we can use such an approach. And for that, we really need large number of samples. So for this purpose, there is a global initiative called the COVID-19 Host Genetics Initiative, which brings together the human genetics community to generate, share, and analyze data to identify genetic determinants of COVID-19 susceptibility, severity, and outcomes, with the hope that such discoveries could help to generate hypotheses for drug repurposing, identify individuals at unusually higher low risk, and contribute to global knowledge of the biology of this infection and disease. So these are basically major three aims of this host genetics initiative. And until now, there are 177 studies have been registered, and these studies are all coming from many parts of the world. And interestingly, we also have three regions from India, Kolkata, Maharashtra, and Telangana, where we have three studies registered in this initiative. So it will be interesting once we have the results from all these studies to sort of compare and understand whether there are population-specific genetic variants contribute to these differences in severity and susceptibility. There are really common factors, common genetic factors that that are present everywhere in the world. So, but we do have some, let's say, intermediary results from this initiative. So for this analysis, they used around 1,300 COVID-19 cases and population-based controls, and these are all from European ancestry, and the results are depicted in this figure. So this figure, it's called as a Manhattan plot. If you're not familiar with looking at such figure, basically, once you perform association tests across millions of SNPs or single nucleotide polymorphisms in these samples, you will obtain a probability value, p-value, for every SNP. And the negative logarithmic of that p-value is shown on the y-axis, and on the x-axis, the physical location of that SNP is shown. And the red line here, horizontal red line, indicates statistical significance threshold. Because we will be testing millions of SNPs, we need to apply multiple testing correction, and any SNP that crosses that p-value threshold is considered as significantly associated with disease. And this blue line indicates a suggestive association threshold. So as you can see, even after using nearly 1,300 cases and 30,000 controls, we are yet to identify significant associations. So we do see some peaks, like for example on chromosome 14 and chromosome 17, and here and there are some peaks appearing, but still, statistically speaking, these are not significant yet. So what does that mean? Does that mean there are no genetic variation playing a role in COVID-19 disease severity and susceptibility? I don't think so. So for that, I was told that there are many students in the audience, and for that reason, I would like to discuss a little bit more on why genome-wide association study, or GWAS, alone will not be sufficient to fully understand and identify a genetic basis for COVID-19 heterogeneity. So the contribution of genetics and environmental factors vary depending on the trait or phenotype or disease that we are interested in. And depending on this, we group these traits as monogenic or polygenic. So for example, Mendelian diseases such as cystic fibrosis, these are caused by single mutations in a gene called CFTR. And this disease is called as monogenic because the entire disease is explained by a mutation in one gene. So therefore, you have one mutation in one gene that has a very strong effect, which explains the entire disease. On the other extreme, you have many complex diseases or complex phenotypes. For example, height is explained by thousands of genetic variants with very small effects, each contributing to the phenotype, height, or disease. So I consider, probably you will all agree that infectious disease is also one such complex trait or complex disease where you have contribution of both genetics and environment. And depending on the pathogen, the genetic contribution may also vary. So therefore, to identify genetic mechanisms for such diseases, we need to go beyond GWAS approach. So why is that? Why GWAS alone is not sufficient? Now, because it's extremely challenging to collect a large number of matched controls. Now, in any infectious disease, defining someone as control, healthy control, is very tricky, because unless that individual is exposed to that pathogen, you don't know whether that individual is going to be remain as a control or resistant or become a case. So therefore, when you use population-based controls, there is high chance that you're mixing up cases also in a control group. So that is one of the challenges when we consider performing GWAS alone in infectious disease. And the second, let's say, insight that we gained is that these genetic polymorphisms, become functional in context-dependent manner. That means some of these SNPs or variations show effect only when there is a particular trigger, only when there is particular infection, only when there is some pathways activated. So in a simple direct one-to-one correlation, we won't be able to effects. And the third and the most important limitation is that GWAS provides association. We identify many risk loci in our genome which are associated with disease phenotype, but it will not tell immediately which genes are responsible and which pathways are affected, and in which particular cell type these genes are activated or affecting the disease. So this remains as entirely a black box. Now, because of these challenges, over the past 5 to 6 years, we have been applying systems genetics approach, and we consider this as a complementary approach to GWAS, where instead of correlating your genome to the phenome, we bring in many layers of disease-relevant molecular responses, and we construct this network to explain the flow of biological information. So how exactly genes, in other words, DNA variations, affect these molecular layers. So if a particular gene codes for a protein, it needs to transcribe into RNA, and this RNA is translated into protein, and some of this protein may work as an enzyme to regulate metabolites. So if we can connect this flow of information, so in a disease relevant in a context specific manner, so then we should be able to not only identify the SNPs that are associated with the disease, but you can also able to explain how exactly these SNPs are affecting the disease. Are they affecting gene expression or protein abundance, regulating the metabolites? You can really construct these networks, which provides much more mechanistic insights than simple correlations. So coming back to COVID, what we know is that excessive immune response, or in other words, cytokine storm, which is also one of the phenotypes in sepsis, is considered to be responsible for severe outcomes. So, if you look at in the later stages, you can clearly see that an overly active inflammatory response is likely to contribute to tissue damage and systemic effects in COVID-19. And this may be related to genetically determined differences in inherent individual immune response mechanisms, as well as being subject to the influence of concomitant diseases. Now, if you think about it, immune response is actually at the heart of many human diseases. So when you have extremely overreacting immune system, you may become susceptible to plenty of autoimmune diseases such as rheumatoid arthritis, multiple sclerosis, celiac disease. On the other end, if your immune system is underreacting, immunodeficient, then you're extremely susceptible to infection. So our body has an intelligent and dynamic way to attain a balanced immune function. So we need this normal immune response. So which factors actually regulate our immune system? Now, about cytokines. So these are the proteins released by our immune cells, and these are key messenger proteins involved in regulating cellular communication and orchestrating both innate and adaptive immune responses. So this is a very simple scheme just to show their importance in response to many different types of pathogens. Our innate immune cells, such as monocytes, may produce a lot of different cytokines. For example, IL-6, IL-1 beta, and these cytokines then try and communicate with, let's say, T cells to initiate adaptive immune response. So all this regulation happens when these cells sense the infections, any foreign antigens. So now we can ask an important question. So do we see differences in immune response in COVID-19? Yes. So if so, which factors contribute to the inter-individual variability in cytokine production, and how do we identify them? Now, to answer this question, so we took a functional genomics approach, where we studied the immune response variability in a Dutch population-based cohort. So this cohort includes 500, randomly selected individuals. So from these 500 individuals, we went on to profile many different things. And most importantly, we quantified cytokines, taking their blood and also taking only PBMCs and isolating only macrophages, and exposing these cells to multiple human pathogens and also toll-like receptor ligands, basically to activate these cells, and after 24 hours of stimulation, several cytokines are quantified to which are representative of monocyte and T cell function. So basically, I'm summarizing the results from 3 different studies that were published quite some time ago to show that many host non-genetic factors also affect cytokine production, and we see some effect of gut microbiome as well on the cytokine production. But I want to focus a bit more on genetics, because what we observed is that depending on the stimulation, we saw host genetic variation explained different proportion of variation in cytokine production. So this is a figure that again summarizes how much of the variability in cytokine production is explained by genetics. And if you take one example, for example, MTB, Mycobacterium tuberculosis, when we stimulate PBMCs using MTB and quantify IL-1 beta and test how much of these differences in IL-1 beta production is explained by host genetics, nearly 100% of these differences is explained by genetic variation. Whereas some other cytokines, for example, if you go for interferon gamma, there is only 25%. So you can see that it is extremely variable, but overall we see a very strong effect of host genetic variation on influencing cytokine differences, cytokine production capacity. So this has implications. So, for example, if we identify a particular SNP, single nucleotide polymorphism, that is affecting, let's say, IL-17 levels in response to Candida albicans, and an important fungal pathogen. So you can see that there is a clear correlation between the genotype, whether individuals have GG genotype or AA genotype. You can clearly see very significant difference in the production of IL-17 cytokines. So we used this approach and identified, hundreds of SNPs in our genome that can affect cytokine production when you stimulate these cells with a pathogen, and then take those SNPs that affect cytokines and test for which disease these SNPs are important. We see clear enrichment for these SNPs to explain many infectious diseases, suggesting, context-specific, identification of context-specific SNPs are very much needed to understand underlying genetics behind these diseases. So this is an example where we applied this method to fungal sepsis. So basically, we used a fungal sepsis cohort. We performed genetic association, and we used this 500 healthy Dutch individuals and looked at Candida-induced cytokines and identified hundreds of genetic variation affecting cytokines. And we performed integrative analysis to basically prioritize genes and then the pathways that are important for fungal sepsis. And these results are now being taken up for clinical trials to test whether targeting these pathways would help fungal sepsis patients. And another example of an approach where you can also use gene expression in addition to cytokines. So if you perform stimulation using, let's say, Candida in this case, on PBMCs, and then you isolate RNA, perform RNA sequencing, and you can correlate the gene expression with SNPs. So here you can clearly see that in normal condition, you don't see any difference between 3 different genotypes. So the expression level of a gene called LY86 is similar across all individuals carrying different genotypes. But when you perform stimulation, only after stimulation you see the differences, become very significant between genotypes. So as you can see, individuals carrying AA have somehow downregulated the expression of this gene. So then you can take such SNPs and test whether they're important in disease. That's what we did here. And as you can see, clearly this polymorphism is significantly associated with fungal sepsis, also called as candidemia. And we went on to show by single-cell RNA sequencing that this gene is expressed specifically in monocytes and B cells, and it also co-expressed with the CCR2, indicating a co-regulation between LY86 and CCR2. And we did a lot of other functional studies by knocking down LY86 to show that this gene is involved in recruiting monocytes and TLR4 pathway. So therefore, defect in this pathway leads to increased susceptibility to fungal sepsis. So in another study, here we used TB meningitis patient cohort from Indonesia. So here we used cerebrospinal fluid collected from cerebrospinal from these patients, basically to measure a lot of things, including cells and cytokines and metabolites. So we quantified hundreds of metabolites in CSF and basically performed differential abundance analysis. As you can see in this map, there are hundreds of SNPs showing difference between controls and patients, and out of these metabolites, we found tryptophan to be significantly different between controls and patients. And when we tested the levels of tryptophan with survival, we clearly saw association of high tryptophan levels with increased mortality, whereas low tryptophan levels suggested improved survival. So then we asked the question whether genetics play a role in explaining these tryptophan levels. So we mapped all genetic polymorphisms in our genome against tryptophan levels in these patients, and we identified many regions in the genome that affect tryptophan levels in cerebrospinal fluid. Depending on the genotype, you can actually see difference in levels. And now we took these SNPs and tested whether we can predict someone to have higher risk for mortality. And we validated an independent cohort that these SNPs actually predict mortality in an independent group of TBM patients. So these are some of the examples that I wanted to show that applying these functional genomics approaches, we will be able to not only identify genetic risk factors, but also explain the mechanism. Now, in COVID-19, we know from many studies that host inflammatory response is extremely excited, and we have cytokine storm. So can we try and quantify what kind of inflammatory proteins we see? So for that, we performed a pilot proteomics study using a Dutch patient cohort. So here we used nearly 120 samples. So this is a prospective study. Right now, we have around 500 patients, but this data, we analyzed in a subset of patients. We quickly wanted to get some insights. And these are just demographic details. As you can see, we have patients who are admitted to ICU and non-ICU, as a severity measure. So we used a proteomics approach. It's a technique called O-link. So basically, we can measure lots of proteins just by using just 2 microliter of serum or plasma or any biological fluid. So it essentially make use of antibodies, connected with probes, DNA probes. So you will be able to amplify these reporter sequences by proximity extension assays, and then you can, quantify each protein using real-time PCR. So, and this technique has, I think, panels for nearly 1,200 proteins or so, and each panel consists of 96 proteins. And we use 3 different panels, so more or less around 240 proteins were quantified in 20 patients who were admitted to ICU because of severity, and 28 non-ICU patients who were in the hospital but not so severe. And basically, we try to look at the different proteins, that may explain the severity, and we find many proteins being downregulated and many proteins being upregulated, depending on the ICU or non-ICU. And in ICU patients, as you may have already seen in other studies, we saw increased inflammation consisting of IL-1 and IL-6 pro-inflammatory pathway, a lot of chemokines, and also there are cell survival and apoptosis factors. And if you look at the genes that are downregulated, you see the genes that are important for hematopoiesis and lymphopoiesis, where you need these genes to increase turnover of immune cells, are downregulated. And kallikrein-bradykinin system, which is an important degradation pathway in lung, which leads to fibrosis and severities, those genes are also downregulated. Now, we can then take those proteins and try and ask whether these proteins are able to classify patients based on severity. And without going to further details, you can see that many of the ICU patients are clustered in this big cluster, and non-ICU patients are clustered on the other cluster, essentially showing, using such markers, we will be able to predict the severity. But the issue is, the response is extremely variable. So if we take IL-6 and look the abundance in ICU and non-ICU patients, you clearly see the average levels are significantly different. But if you look at the spread of the data, you see that there is extreme inter-individual variability, right? And the other point I also want to make here is that if you now measure this IL-6 at different time points once they're admitted to ICU, nearly, these are patients in ICU, nearly 30% of these patients in the second time point show less IL-6, very similar to non-ICU patients. Does that mean, the response can vary in these patients when you give some time? And what factors are actually contributing to this? Does this also contribute to reduced severity when you allow? So these are all some of the questions we can ask if we can measure such data in large number of samples. So for that reason, now we are measuring gene expression, a lot of different proteins and metabolites in this patient cohort, and with the hope that we can then perform system genetic studies. And hopefully I can share our results once we are done with that study. And very quickly, I want to ask a question: is blood sufficient to identify all the factors? Because in sepsis, we know that just measuring cytokines will not be able to explain the molecular mechanism, because there are so many other responses are involved in determining the heterogeneity in outcome. So in sepsis, what we know is that when there is an infection in the blood vessel, cytokines produce a lot of cytokines, and these leukocytes produce a lot of cytokines, and these cytokines then affect endothelial cells, which lead to leaky vessels, organ injury, and death. So therefore, people have tried for many years blocking inflammation with the hope that this will help sepsis patients, but all the clinical trials have failed, and we still do not know how to explain, how to predict the severity in sepsis. So I think we are ignoring an important component here is endothelial cells. And we performed the study and showed that even if you perform stimulation of PBMCs or blood cells with different pathogens and look at gene expression, all these genes somehow converge on endothelial cells. And also from genetic study, we have shown that all the genes that are associated with fungal sepsis are enriched for endothelial cell pathways. So I quickly want to make the point that endothelial cells also contribute to overall inflammation in sepsis. So for that, we performed an in vitro experiment where we initially stimulated PBMCs using sepsis-causing pathogens, and then we took the supernatant and added on the endothelial cells, and then we looked at endothelial cell response. And what we found was that even if you block IL-1, TNF-alpha using blocking agents in the supernatant and add that supernatant, which does not have the effect of IL-1, TNF-alpha, these supernatants still able to activate plenty of genes in endothelial cells, and those genes are enriched for interferon pathway. So suggesting an explanation, giving an explanation why if you only block IL-1 and TNF-alpha, there is still increased inflammation in sepsis, is because the other cell types involved in contributing to this inflammation. So therefore, also to study COVID-19 disease and the viral infection, respiratory viral infections, we need a disease-relevant model. Just using one cell line or looking into biopsies or organoids may not be sufficient. And we are, for that reason, trying to make use of organ-on-a-chip technology, because modeling the complex biology is extremely difficult in cell-based system. So this tiny chip can recreate the smallest functional unit of an organ, enabling us to more easily and accurately study the inner workings of biology underlying COVID-19 disease. So let me introduce the organ-on-a-chip that we are using. So if you take a cross-section of the chip, you can see a bottom channel and also a top channel. So in a smaller these two channels are completely separate and separated by flexible porous membrane, with like 7 microns in diameter, these pores. So we can basically seed cells on either side of this membrane. Usually epithelial cells on the top channel and endothelial cells in the bottom channel. So we can then flow media independently through these two channels. And if we need, we can then replace the media with air for lung epithelial cells. So we can also add immune cells to the chip, for example, to study cytokine chemokine release, which is needed to recapitulate the clinical phenotype. So we already have this system, and we are able to grow endothelial cells and epithelial cells, co-culture these cell types on this system. And we will be performing experiments using this system to understand how exactly, this virus contribute to different outcome in different patients. Because the one additional advantage of this system is that we can use induced pluripotent stem cells to derive patient-specific endothelial cells and epithelial cells, so that we can clearly study how having a particular genetic background affects cellular interaction and thereby contributes to overall inflammation. So to conclude, we have been using functional genomics approach, where we are making use of both patient cohorts and population-based cohorts, and performing stimulation experiments to obtain insights into genes contributing to the disease, and applying organ-on-a-chip technology and other functional work to understand and gain insight, so that we can then use this knowledge to predict and develop novel therapies. So with that, I would like to close this webinar, and I'm very happy to take any questions, and thank you all for your attention. I also would like to thank all these people who contributed to our studies. Thank you very much.
No, you don't do it. No, you don't.
So thank you very much, Vinod, for this excellent talk. I think it was very, very informative and enlightening. And you have presented so much of data from your ongoing studies, and I'm sure there will be more coming as your studies are completed. And we are happy to also see that your study also involves some sites in India. There have been a few questions which have come up from the audience. I'll take some of them which are, I think, directly related to your topic. One point is that we hear about the virus undergoing mutation. Being an RNA virus, it undergoes rapid mutation. Now, among these different genetic variants of this virus, are there differences in the cytokine response in the host?
So, yeah, thank you. That's an excellent question. So, indeed, I focused only on host genetic variation. So, as in one of my introductory slides, I did show that we should consider pathogen factors as well. Absolutely, depending on the and the virus strain, virus genetic background, our immune system also respond differently. However, for now, of course, here we're going to use a strain that is more prevalent in Dutch population, but I don't think anybody has looked at differences in immune response based on different strains of virus. But that's something we should definitely consider doing. And I guess when people do such studies in different countries, they are going to use the prevalent one in those countries. So hopefully those strains will also become available so that we will be able to include a battery of viral strains to screen and identify if there is any difference in cytokine production depending on the virus strain.
Yeah, another question is related to the relation between age and disease susceptibility and gene expression. Now, of course, you said the main pathology is related to the immune response.
Right.
And is there any possibility of, say, downregulation of genes as a therapeutic approach so that we can the immune response?
Yes. So that's also very good question. So there is very recent study published in Cell showing this virus compared to influenza or other types of respiratory viruses. This is strange in a way. It hijacks our immune system to suppress type 1 and type 3 interferon response. So if you look at other viral infections, so interferon response is the, let's say, first line of defense, which become active and be able to then initiate other adaptive immune responses. But in case of COVID, this coronavirus somehow suppresses the interferon response and immediately activates the adaptive immune response. So therefore, we see very strong cytokine storm. So looking at that, it is feasible that if we can downregulate some of those viral proteins important in suppressing our type 1 interferon response, can be potential therapeutic angle. So if I'm not mistaken, I think remdesivir is one of such drugs where it tries to also improve interferon response. And of course, people are now considering running such clinical trials.
Yeah, another question is with respect to ivermectin, which is been approved by FDA. Do you have any comment on this and how it affects the immune response? Is it through immune-mediated action of ivermectin? No.
Oh, sorry. I will have to say I don't know much about that particular drug right now.
Okay. Okay, and one of the participants wanted to know a little bit more about this chip that you talked about in the before one slide. Can you,
Right.
Yeah.
So this is so thin that you can actually hold in your hand. So you can compare it to a microscopy slide that we use for, let's say, doing staining, and it, as I already said, so this is embedded with microfluidic channels. So it contains 2 channels separated by a porous membrane made out of material that supports the growth of human cells. And it's from a company called Emulate. Basically, these organon chips are becoming very instrumental in understanding biochemical processes in a very tissue specific manner. So this needs a container, also called as a pod, with it's a reservoir where we can put cell culture medium, and these reservoirs can be placed in machines that are placed in incubator. These are called Zoe. So it basically regulates fluid flow. So you can also adjust what flow rate you want to have, and also carbon dioxide levels. So once you culture the cells, so you can use these for, let's say for not only infecting with virus and looking at cell-cell interaction, you can also use this for screening drug effects. For example, you want to apply a particular drug and see whether it helps for monocytes to adhere much more efficiently to endothelial cells than other drugs. And you can also take these cells, isolate RNA or protein, and do all omic type of analysis. You can cut and perform histopathology to look at proteins. So it is quite flexible in that way. And right now it is still an expensive experiment because the chip is quite costly. But when we go to specific patients or to screen for specific drug effects, then this becomes extremely handy. So I don't know if there is any specific question on this apart from this general information.
Yeah, maybe the participants can interact with you later. We can share your email, and questions can be passed on to you. And there's one more question on what factors are produced by these endothelial cells as a part of this response here.
So so in case of sepsis, we have seen that endothelial cells also produce cytokines and many other chemokines, and so endothelial cells also produce our cell surface proteins such as ICAM and VCAM, which are important for monocyte adhesion. So in COVID-19, we speculate that interaction between during the interaction between immune cells and endothelial cells, and again epithelial cell as well, the endothelial cells also produce ACE2, which is a receptor for COVID-19. So the severity can also be influenced by the responses from these endothelial cells. So therefore, we would like to apply such an approach so that we can then separate these cellular components after performing infection studies and look at transcriptional response and other let's say omic layers.
Thank you, Vinod. There is one question for which I would also like clarification. Now, just as you do organ chip study, taking any tissue into consideration, have you thought of looking at all of these system-based approach with respect to tissue? For instance, the receptor binding domains will be distributed along a particular tissue, but in pockets, okay? Mm-hmm. So even in a particular tissue, there may be regions where these domains are there and regions where it's not there. Would within the same individual, picking up areas which represent, when you make a chip, tissue-based chip, is going beyond organ chip, tissue chip. Mm-hmm. Do you think you could look at the areas with these domains and their responses, and to areas where they don't have domains and the response, because that will tell within an individual, apart from the quantification of the domains that are available, what is the difference? And if you have to look at, receptor binding that is being talked about, mm-hmm. Would your kind of strategy help?
So within a given organ, whether different tissue,
say endothelial tissue, when you see,
right, yeah,
you have picked up endothelial tissue, and you know that the receptor binding domains are located along the blood vessels, right?
Right.
So could there be areas even within the same individual, because you are looking at genetics as well, and mm-hmm. therefore within an individual, if you pick up areas where these domains are located and where it's not there, would it help to look at differences?
Absolutely. This is also an excellent point. So in case of, let's say in intestine, such studies have been done, may not be already with COVID, but let's say when it comes to inflammatory bowel disease, although it's one organ, depending on the tissue component, the gene expressions, even in one individual, the gene expression, if you target one particular gene, the expression of that gene can be very different in different components. So yes, we can apply this methodology to answer that question, and then we can perform, let's say, single cell RNA sequencing, and now methods are also being developed to look at single cell other omics as well. So when we do that, we can then trace back, even if we collect epithelial cells, endothelial cells from different locations, we can trace back how this gene expression pattern or any other response is different to different domains of these components. Yes, that's feasible, and I guess that probably will also answer important questions. Yeah.
So there are some more questions, but as we are running out of time, we now have to wind up. We thank you a lot for having accepted the invitation, I should say, at a short notice. On behalf of Nitte, Nitte University, and of course the Center for Science Education and Research, I am extremely grateful to you as well as to our Vice Chancellor for having permitted us to have this webinar at a very short notice, which is why there was some glitch in the beginning. But I am looking forward to go back and read a little bit about what you have spoken, because it goes way beyond our own knowledge at this stage. But then when you have your kid doing very, very well in science, you're very proud of that. And therefore, we are, I should say, extremely, I wouldn't like to say grateful. We are delighted to have had you. And we do hope that this will be the beginning also of collaborations that were supposed to, of course, happen. But due to COVID, we have just kept it in abeyance. But it will happen. This is not going to last. It will definitely be solved. I will now request our Honorable Vice Chancellor to give his final remarks before we wind up. Excuse me, sir, can I have you say something? Yeah.
Thank you, Vinod, for that wonderful talk on role of genetics in COVID. As Indrani said, it is really enlightening, but our knowledge regarding genetics, particularly molecular genetics, is not all that great. But definitely we understand that it was very enlightening for our people who are working in the NUCSER, and other laboratories of the university who are working on COVID. Particularly, recently we had opportunity to equip our lab with the molecular genetics facility for COVID testing, and is about to be started, and we have got all the equipments. I think that will definitely add to the research facility in the university. And as you know, we had one patient, COVID patient, 10-month-old child, which got treated successfully and got discharged. And one question I wanted to ask, why the children are spared, even though their immune response is poor, why they are spared, particularly mortality is less, I was told, in children. Why? Could you just enlighten on this point?
So, sure, sir. First of all, let me quickly thank dear madam and sir, for talking such nice words about me. So the idea was just to give an overview on different technologies and approaches that are out there, and it's really not my intention to complicate the concept, but to I heard that there are a lot of students, so hopefully for them it's good to see what can all be done using such techniques. And thank you, Dr. Satish Bhandari, sir. So to come back to your question, I think ACE2, we talked about ACE2 expression. So it's one of the receptors for this virus. So this looks like the expression of this receptor is highly variable depending on our age. So somehow the mucosal surfaces in children are not expressing this receptor as strong as elderly people. Of course, ACE2 has other function. So when you get old, you need that protein to keep the respiratory system stable. So therefore, you need that protein. But in children, there seems to be lower expression of this receptor. So there is restricted viral entry. But this is only after maybe 7 or 8 years of old. If you go less than 5 years old kids, this is equally severe in small kids as well. But of course, more studies will provide much more insights into these differences.
Thank you, Vinod, and congratulate Indrani and Idya and Anirban for organizing this webinar on again. We had one in the morning, and this one more in this related subject. Thank you very much.
Thank you very much, sir. I'm really looking forward to visit, with our colleagues soon, the Nitte, and we should definitely initiate collaborative studies. Thank you very much.
Thank you. Good day to you. Pleasure having you. Okay.
Thank you.
When Anirban said he contacted, we were delighted actually. Okay.
Thank you.
We will have more as your
Definitely.
research.
I'll be happy to.
Different areas. I know your own infectious disease area is very strong. So we will like your sepsis award lecture to be delivered to all of us here. Anusha heard, but we all have to also hear. That is why. Okay. Thank you very much. Thank you.
Yeah. Take care. Bye-bye.
Bye.